Hello everyone, I'm Dr. Venkata from School of Chemical and Biotechnology, and I welcome you all for another virtual lab session in Drug Design Laboratory course. In this session, we will look at the experimental technique called isothermal titration calorimetry, or simply called ITC. So this is an experimental method to determine the binding constant of a protein ligand complex. Okay. In this session, I will discuss about the instrumentation, experimental setup, data analysis, and applications of this technique. Okay, so we already know about protein in protein ligand interactions. How a protein binds to a ligand, that is, it binds to its active site or binding site through a weak non-covalent bonding interactions such as hydrogen bonding, hydrophobic interactions, van der Waals interactions, etc. Okay, these interactions stabilize the protein ligand complex, and thus gives them a lower free energy than the individual energies. And we've already learned to perform molecular docking, in which is a method, which is a computational tool to predict a preferred orientation of a small molecule that could potentially bind to a protein molecule, a receptor molecule, and we will also be able to predict the strength of association. In other words, quantify the binding affinity using some sort of scoring function depending upon the type of program that we use. So there are various programs that give you different types of the constants, scoring values, and so on. However, all these methods are theoretical, give you a theoretical binding affinity. However, it is very important that in the drug discovery process to validate these findings. Okay, so this can be done by performing various experimental methods that can be used to determine the binding affinity or the binding constant of a specific ligand to a protein. Okay, and in drug discovery world, we usually refer to this binding constant as IC50. Okay, there are many experimental methods available that can be used to determine binding constant of a uh, protein ligand interaction. So there are many spectroscopic methods that can be used. Use of, for example, fluorescence spectroscopy, UV visible spectroscopy, CD spectra, and we'll also use NMR spectra. So all of them uses variation in absorption or emission based on the chromophore or fluorophore. So depending upon that, all these things requires some sort of labeling, and often they have varied sensitivity. Okay, so in addition to that, we can also have a specific binding assays, enzymatic assays such as ATPase assay or malachite green assay, phosphate assay, and so there is many such assays are developed for a specific class of enzymes, and they can be used to determine the binding affinity by performing kinetic experiments. In addition to that, we can also use ultra analytical ultracentrifugation, stopped flow methods, and also recently the surface plasmon resonance methods. Okay, so all these things again can be used and offer varying levels of sensitivity, and all of them could be able to give you the binding constants. The method that we are going to study, that is isothermal titration calorimetry, comes under the microcalorimetric methods, and in this method we measure the heat of binding. Okay, and this is a highly sensitive method. Okay, ITC has many advantages over the other methods. Okay, so this does not require any labeling of the samples that is required for fluorescence microscopy or NMR spectroscopy, and this one shows high sensitivity. That means that even a much weaker binding ligand or the very weak KD values can also be determined, and it is highly accurate, and it also allows us to measure many thermodynamic parameters also. And ITC is the only method that can measure the binding energies directly of either a protein ligand binding or a normal small molecules binding. All right. So it measures the key heat change associated with the protein interaction with its ligand at a constant temperature. As the name isothermal says, that the experiment is performed at a constant temperature, and hence the whatever change that is happens has to be measured at a constant temperature. So during the interaction of a protein with its ligand or an inhibitor, depending upon the type of reaction, it might either take up some heat or it might release some heat, depending upon whether it is an endothermic reaction or an exothermic reactions. Either it is release of heat or absorption of heat. Both of them will be measured accurately by this method. Okay, and this method provides us various thermodynamic parameters such as enthalpy, entropy, and so on in a single experiment. Okay, so what are the parameters that we can get out of ITC experiment? So we can get parameters that would help us identify the strength of the interaction, that is the delta H, that is change in the enthalpy, and also the binding affinity. So these two directly let us know about the strength of the interaction or the efficiency of the binding. So we could also get the delta G value, that is the Gibbs change in the Gibbs free energy, that would talk about the overall stability of the biological complex, whether the new formed complex is stable or how stable it is in relation to the reactants, the unbound structure, and so on. So those things can be easily measured. So basically, lower the delta G value, that is the free energy, the higher the delta G, that is high negative value, means that it has much higher stability, and so on. It also gives us the measure of change in the order or disorderedness. So how efficiently, how ordered, or how flexible the protein inhibitor constant, the complex, by measuring the delta S, that is the entropy change of the binding reaction. So one of the important parameter that is often not can't be determined by the other methods is the number of binding sites. Often the proteins have just one binding site and binds to the ligand in 1-to-1 ratio. However, there are certain proteins, there are many proteins nowadays that have potential to bind more than one binding site. So that can be easily identified by this n value, that is the stoichiometry of binding. So if the stoichiometry is 1, that means that you will have just 1-to-1 complex, that means that there is only one binding site. And we have examples where there is more than one binding sites also, that means that we will have the stoichiometry of 2. Okay, so that's telling us that there is 2 sites in the enzyme that we should be aware of. All right, so in addition to these information, so ITC also sheds lights into the mechanism of inhibition or the interaction. So they gave more insights into the binding energies, how it is done. Okay, so as I already said, that it gives you the values of delta H, delta S, and delta G. In other words, it gives the relative contribution of delta H and delta S towards a delta G. Obviously, delta G has to be negative for a spontaneous process. That means that delta G values always should be negative. But however, the delta G is given by H plus T delta S. Okay, so delta H minus T delta S. So the delta H and delta S can have different values and different signs also. Okay, so that means that based on the values of delta H and delta S, we may be able to get some insights into the type of interactions that may occur in the protein inhibitor protein ligand complex. All right. So in general, the delta H gets values or depends on the number of bonding interactions, such as hydrogen bonds or van der Waals interactions, and so on. So these type of interactions are reasonably strong and contribute strongly towards the binding affinity and or enthalpy driven processes. Okay, however, they don't enhance rigidity. So the contribution from delta S are usually associated with hydrophobic interactions, relates to the increasing or decreasing the number of microscopic conformations or configurations, okay, that might arise due to hydrophobic interactions, and that has directly linked with the flexibility. So if it has more hydrophobic interactions, that means that the ligand is going to be more rigid and it is going to have less flexibility. All right, so that is going to have high delta S values. If in the absence of many hydrophobic interactions, the ligand is still going to be comparatively flexible, and hence it may not have a large delta S value. So the contribution will be hugely from delta H values. So these are highly empirical conclusions that definitely would give us heading to understand the type of interaction that we would require. So to understand this better, let us do an illustration with a typical example. So let us take three compounds, that is A, B and C, that has same affinity but different energetics. So you have all three of them. So you can look at the delta G value, you can the blue bar. So you can see all of them have the same value, that is the overall energetics. So that has same value. However, when you look at the red and the green bars, that is contributing to the delta H and the T delta S. So you can see that they are different in all three of them. So even though when you look at it, if you do any other experiment, then you're going to have same KD value because the delta H is the same. So that means that the values are going to be exactly same. However, their contribution is different. So from this value, so let us see what we can conclude. In the first one, that is A, you have delta H is it has highly favorable, and the delta S is not so favorable. In B, delta S is favorable, that means it has a high negative value, while delta H is not so much. It has a slight positive value, that is in the unfavorable region. In the example C, both of them contribute positively on the same side, so everything is favorable here. Okay, from this what I can conclude or for what I can hypothesize is that the first compound, that is the A, it has a lot of good hydrogen bondings that gives you a lot of binding energy. However, the conformational flexibility is still there. That means that it could have more multiple conformations that could still be there. So it means that the conformational rigidity is not there. On the other hand, the second case, the binding is predominantly hydrophobic in nature, does not form any hydrogen bond or van der Waals interaction, any such that. However, it has have a strong hydrophobic interaction, such like that. Okay. In the third one, also it does have both hydrogen bonds and van der Waals interactions, and probably it also has a preferred orientation. Okay, so that like that I can say that this B is predominantly forms a hydrogen, hydrophobic interactions, and probably does not have any hydrogen bonding interactions, or it is not in preferred orientation. Thus, maybe I would be able to, by introduce hydrogen bond by certain modifications may help the affinity and so on. So, like this, we'll be able to get some understanding as to the mode of binding of a ligand. So with that introduction, let us go on to see how the instrument ITC experiment works. So the instrumentation, this is the schematic diagram of an ITC instrumentation. So it contains two cells. So one is a sample cell and the other one is a reference cell. So both the cells are tiny cells. Depending upon the type of ITC that we work, the volume changes and the the material changes. So you have a sample cell and a reference cell. Okay, both are made up of highly non-reactive metals. In other words, obviously you don't want the cell reacting with the substance, the reactants, and also they should be highly efficient thermal conductors, because we are measuring the heat energy exchange or release. So we need to look at the cell material has to be highly thermal conducting. Okay, so the materials that are commonly used are Hastel alloy in case of nano ITC, and then you can also use gold. Okay, and both of them are enclosed in an adiabatic jacket. So that means that this enclosure is completely heat proof, so that no amount of heat can enter or exit from this thing. And then these two cells are coupled with a powerful thermal coupler that could maintain the temperature between these things. So both the reference and sample cells are maintained at a constant temperature. So there is the same temperature. Okay. Then in the sample cell we add the protein protein solution in a buffer, obviously, and in the reference cell we just include buffer as a reference. Okay, so now both the temperatures and both the cells are maintained at a constant level, the same level. The delta T is maintained as 0. Okay, and then we inject the ligand using a micro syringe into the sample cell, at regular intervals, and a specific amount is injected at regular interval. All right, and there is a constant stirring is also maintained. So upon addition of injecting the ligand, so let's say if I inject, let's say, a couple of microliters, then that is going to cause that is going to react with the protein, and during this process there will be heat evolved or taken up, depending upon whether it is endothermic or exothermic. Then there will be either absorption of heat or release of heat. So that release or absorption of the heat is measured by the thermocoupler. So basically how it functions is that when the heat is released, then that means that the temperature of this cell is going to go up, and then obviously the coupler tries to adjust the temperature by absorbing the heat. So the amount of heat absorbed by thermal coupler is measured in terms of microjoules, and if it is released, then it is same is measured by the same setup. Okay, so here let us take schematics. Then let's assume that this is a protein molecule, and this is just a ligand molecule, and this is protein ligand complex. During the first injection, obviously before the injection, the cell contains only the protein molecules, and upon injection ligand, so there are few ligand molecules enters into the sample cell, and then they interact with the protein molecule, forming the protein ligand complex, during which either heat is emitted or exchanged or absorbed. So let us assume, for example, that it is emitted. Then that means that obviously the temperature rises, and then the thermal coupler tries to adjust the temperature by absorbing the heat. So we measure the amount of volt, the energy that is supplied to bring back the temperature to the earlier state. So that is what is measured here. That is one injection. You can see there is a change in the temperature, and then it is brought back to the same equilibrium. So that is one equilibrium. Once it reaches the equilibrium again, then there is a second injection is injected. Then obviously the same process happens. Okay, so and then it is continued. As it continues, then every injection there is more and more inhibitor molecules is introduced into the sample cell, and then you can see for at every injection, then it is allowed to equilibrate. That means that we have to get back to the normal baseline value. Then before we start the next injection. So you can see that amount of heat required sort of increases over the subsequent injection. After the completion of titration, you can look like a sigmoidal curve. Okay, so it just keeps on increasing, and towards the end there is no or very minimal heat exchanges mentioned. So why does that happen is because the ligand is usually present in much higher concentrations. So when you inject it, and then the more and more protein molecules are saturated with the ligand, and hence at some point it is fully saturated. After that there is no heat is released, because there is no more binding, because all the enzyme molecule that is present in the sample cell is already bound with an inhibitor molecule or a ligand molecule. Hence any additional injection of ligand would not contribute towards any release or absorption of heat energy. Okay, so as I said earlier, the way the measurement is done is the amount of power is required in terms of microcals per second or microjoules per second to required to re-equilibrate the temperature in the sample cell. Okay, the heat exchange is then simply calculated by integrating the heat power over time in seconds. Okay, it is measured over time. Okay, so this is the typical data that we require, that is how much microcals per second that is has been required against the time plot. So then each and every spike is refer to a specific injection. So usually typically we do somewhere between 20 injections, and we can see there's so many different curves. Okay, so this one is converted into this plot. So we plot the molar ratio of each and every injection. That is, you can just say it is injection or molar ratio. Okay, so the molar ratio is plotted against the area of each and every curve. So we calculate the area of the curve, and then we integrate, and then we will get something like this. Okay, so this is the plot. Okay, so we use non-linear regression to plot to fit a curve in this one, and obviously the parameters that we could extract from this plot is the height, that is equal to enthalpy change, and then the slope is the affinity constant Ka, and the midpoint is the stoichiometry molar ratio. If it is 1 to 1 or something like that, then we will get this one. Okay, from these values, these three values, we can extract the other thermodynamic parameters, because I said we could get delta S, delta G, and all. So we have, from this graph, we only have the height, that is the delta H value, that is we already know, and then we also know the Ka. So there is a difference between Ka and Kd. So Ka is the association constant and Kd is the dissociation constant. They are reciprocally inverted. Ka is equal to 1 by Kd and Kd is equal to 1 by Ka. So depending upon what is your requirement, we will be able to get this information. Okay, so and also be mindful of the fact that which one that we actually get out of the solution. All right, so the delta H value can be calculated using this expression. Delta G is equal to minus RT ln Ka. So this is something that is thermodynamic from second law. So this is something that we already know. Then we also know the Ka value from this plot. So we can calculate the delta G value. So we can calculate the delta S value from this equation. So that is delta G is equal to delta H minus T delta S. So in this equation, we know the delta H value, and then we also calculate the delta G value. From that, I will be able to get the T delta S value, or just also delta S value. Obviously, we know the temperature, that is already we fixed the experiment. Okay, so there is one small experimental setup that we have to pay into attention is depending upon the molar ratio, that is the concentration range, you might just end up getting this part or this part. So like that means that you would have different types of curves that might be getting. So for an ideal solution, that means to get the actual values of the Ka midpoint and so on, then we should have a curve of this type, that is your sigmoidal curve. So we cannot have a curve like this, or we cannot have a curve like this. This is too steep, that means that the molar ratios are too narrow, that means that it's going to have like an a step curve. Or if it is too too broader, that means that you're going to have a linear. That means that I'm just looking at just a small part here, so that means it's going to be linear. Or else if this one is, I'm just looking at the whole thing is compressed into smaller range, so that means that it's going to change rapidly. Okay, so in order to get this thing, because if you either of these types of curve, it obviously you won't be able to measure the slope and the midpoint accurately. So maybe they may give you the enthalpy change to a certain extent, but then not the other two parameters. So we have to get the ideal shape of the curve, that is your sigmoidal curve. For that, in order to get these curves in range, so we use another value called C value. So we need to have a C value at a particular range. Okay, so the C value can be calculated using N, that is the stoichiometry, into Ka, that is association constant, into M, that is the concentration of the protein. All right, so depending upon the affinity values, we have to modulate, because N is not going to change. Again, the affinity is fixed for a protein. The only thing that I can modify here is M. So basically, depending upon the Ka values, I have to correspondingly modify the M value to get a C value in range. In other words, to get a curve of this type. All right. So basically, it means that strong complex, that means that you have a high Ka value, means that you would have a low protein concentration is enough. And while Ka, if it is a weak thing, then weak protein or weak interaction between the protein and ligand, then you need higher protein concentration. That's a rule of thumb. So basically, we have to do a couple of trial runs to get the actual concentration range. Only then we will be able to get the ideal curve shape, and then we will be moving on to that. Okay, so depending upon the type of, we talked about both type of reactions. One is endothermic reaction and also an exothermic reaction. So during endothermic reaction, the heat is heat is absorbed. So the curve would be this type. So exothermic, whatever we've seen is the exothermic reaction. We can see that the heat is released. For endothermic reaction, the curve profile would exactly be the same, but it would be inverted. So that's also one. In one type of reaction, it is released. In the other type of reaction, it is absorbed. So it is just simply a mirror image of each other. Okay. So again, how does a binding and a non-binding looks like? So if there is a binding and it is exothermic, then you can look at something like this. So there is a binding, that is typically what we have seen earlier. Okay, what happens if there is no binding? So if there is no binding, then you'll get a curve like this, because there will not be any significant change in the during the subsequent interactions, so subsequent injections. All right. So So every injection, the only heat change that occurs during the injection is because there is no interaction between the protein and the ligand. So only thermodynamic change, the contributions would be from heat of mixing and heat of dilution. Okay, the heat of mixing is very constant and it is applied across the all injections. Heat of dilution is something that varies to a certain extent. But obviously, when you look at it, it would be like a linear, straight line like this. Okay, so hence we can just by looking at the simple profile, we'll be able to tell whether there is binding or no binding. Okay, so we do have one of the ITC experiment at the Shastra campus, the one that is situated in ASK 1 in characterization lab. We have the equipment is made by TA Instruments, that is called a nano ITC. This is the equipment, and this is what we call as a degasser. This is just use a filter, and then we also have a PC and a printer. So this is an equipment that is available for use for anyone from Shastra University and also outside Shastra for charge. Okay, so let us look at how we will set up this one. Okay, so initially what we have to do is we can just remove the piston head, that is the motor head, and then we can just remove it, and then that will give you access to the sample and reference cells, and you will inject the sample, that is your protein, into the sample cell, and also fill the reference cell with the buffer. Once this is done, then we will take the injector needle, that is a ligand lean needle, that is a micro needle, and micro syringe. We fill the micro syringe with the inhibitor molecule and then attach it to the piston head. Then the piston can go back into the equipment with the needle. So the needle also contains a small stirrer that will also that will be used to stir, and the whole thing is connected to the computer from which all these things, all the parameters and everything can be controlled. Okay, so so before talk about the data, let us discuss about the sample preparation. So all the samples that we use, that is your protein solution, buffer solution, and the ligand solution, needs to be free of any dust or solid particles, and needs to be filtered through 0.2 micron filter. So, and also they need to be degassed, so that is to remove the any dissolved oxygen or air that might be present during your sample preparation, because any dissolved solid particles and these things can lead to generation of air bubbles, and also that could lead to the formation of heat spikes, because this method is highly sensitive and all these samples to be in perfect condition. Okay, so any irregularities in sample preparation can have a significant effect in the data interpretation or the experimental measurements. Okay, so then we add the protein, as I said earlier, we add the protein in the sample cell, buffer in the reference cell, and the ligand in the injection syringe. Okay, then we will set a specific temperature and a stirring rate, that is RPM. So we typically measure them at 25 degrees C, and the stirring is maintained at 200 RPM. And we also designed the inhibitor syringe is about 50 microliters. So we design a specific injection volume and the interval, that is let us say that 2.5 microliters at every 5 minutes or so. So we have to give enough time for the after each injection so that it can come back to the normal base value. So that means the time interval also has to be and has to be maintained. Okay, you don't want to keep it at too long, or at the same time not too short. So you have to arrive at an optimal time length and the injection volume. Okay, so the injection volume has to be we have to take into account, because if there is a weak inhibitor, obviously you need to inject a little bit more so that you will get a measurable or significant amount of heat either released or absorbed. Okay, so if it is too low, again we will have more background noise. Obviously that we have to pay attention to that. Okay, and after setting up all these things, before we start the experiment, that is injections, we have to allow the equipment to equilibrate. Okay, because we added all the samples from various conditions, so you have to allow them to equilibrate, because equilibration takes more time, because all of them sit in an adiabatic chamber. That means there is no heat in or out. So we have to allow them to equilibrate. Even just a 1 degree change would take a good 5 to 10 minutes to equilibrate. Okay, so that is why we have to ensure there is equilibration, that the sample, reference cell, injection syringe, all of them are in same temperature before we actually start the measurement. Okay, so typically we allow about 30 minutes for equilibration. Okay, so in order to we would also have to do a blank with ligand alone, ligand buffer, protein buffer, and buffer. Okay, so why do we need to do this is because the dilution effects, because when you inject a small amount of inhibitor into the protein, then there is dilution takes place, and there is also heat of mixing. So all these things, as this experiment is very sensitive, and depending upon the type of solvent and the inhibitor and the protein molecule that we set, these dilution effects can be significant. So to avoid any errors because of this, we do take precaution and we subtract these dilution effects from the actual measurement. So we have to measure the heat of dilution of ligand and dilution effect of protein and dilution effect of buffer. So all three of them getting diluted, and we can measure them separately. By ligand dilution would be just injecting ligand into buffer, and then protein dilution is just injecting buffer into protein into buffer into protein, and buffer will be buffer into buffer. So all these things can be measured, and we can calculate the corrected heat exchange by using the equation. So we just run the experiment, and then from that experiment measured values, you subtract the heat values of Q ligand, protein, and buffer. So that I will get the corrected Q values, that is the heat exchange. Okay. So let us look at how we can analyze the data that has been derived. Okay, so as I said earlier, the model fitting and everything is done by proprietary softwares that are provided by the manufacturer of the equipment. We do have two manufacturers that provide the equipments. One is TA Instruments, that one we have, and then there is also GE Healthcare now, which is Malvern. So let us look at the Nano Analyze software that is used by the Nano ITC that is sold by TA Instruments. So this is a software that I'm sure you can download. If you aren't interested, you can download the software, which is open source at the moment, and we can analyze the data. So from here we can just extract the data that is derived from the equipment, can be imported into this, and the data would be look like this. So as you can see, the baseline is not quite flat, so there is always be some sort of, because as we adjust the heat energy, as the heat is released every time, and the thermal coupler slightly adjusts the baseline temperature, reference temperatures, and there is always a slight variation to the baseline that needs to be corrected to start with. The first option is to correction of the baseline. So we can use the baseline correction option, that is this here. Just click it, and then you just have to draw a baseline like this. So you can just choose click points, and then automatically then you can just ensure that the blue line that I have drawn is somewhat covers all the bases, and then you just finish. Then what you'll get is something like this. Okay, so this is your baseline. So now you can see the baseline is some more or less corrected. Okay, so at this point we have to do one more thing. So as I said, that we will be able to subtract the heat of dilution of various parameters. So here I can use this method to subtract. So if you subtract that, then it will automatically remove the contribution to heat of separation. So remember that all the other conditions has to be maintained. The same injection frequency, same injection rates, and same volume of injection, everything else has to be exactly same for all the experiments that I mentioned. Okay, so that is the heat of dilution for protein, ligand, and buffer, and for your actual experiment. So all of them has to have same parameters. So if you change any parameter, obviously that will lead to the change of values, and that would affect your data integrity. Okay, so all of them has to be maintained at the same level. So from that, you can see, just go here and then look at the area that will integrate everything. Then you also have to provide your the various concentrations and injection volume, etc. So usually all these parameters will be taken from the input file, because they are already set up from the input file. Okay, so you can see there is injection, each and every injection we did 20 injections, so I can show there are 20 of them. And then you can also say Q is in the microjoules, and then corrected Q, because in this method we didn't do any correction, so that's why it's showing the same. So if you subtracted the heat of dilution, then you'd get this thing. From this, obviously it calculates the syringe concentration, cell concentration, and then the molar ratio, and so on. Okay, so next moving on to the plot, the next plot, that is molar ratio versus the area under the curve. Okay, so this is what you get. Obviously, as you can see that it looks like sigmoidal. There is no issues. We have already got a good starting point. And usually one thing that you have to observe is here that the first injection usually does not fall into place, because usually there will be some kind of, so it is usually because the first time that an equilibration does not occur properly. Sometimes usually the first point would not have the same trajectory. So usually if there is a first point or any point where there is an issue because of air bubble or something, then you can carefully remove that point and then so it's a standard that to have a first point removed. Okay, so that would usually be out of position. Now what I can do is use a non-linear equation or a model to fit this one. Okay, so each and every company, they will have their own proprietary models covering a single site binding, two site binding, or sequential binding, all these type of various possible scenarios. They've created models, and we'll be able to choose just one of them and to fit the data. So this is that I'm just using a single site model, and I just chose them. So basically it just gives you a default values, and now I'm going to use the curve fit option and then fit this model to the data point that I have. So when I fit it, then that will calculate all the parameters, and this is what I will get. So these are the experimental point, and then this is the best curve fit that I have, and it throws me out all these points. So there is a KD, there is N, delta H, delta S, all these values are provided to me by the software itself. All right. So now from these are the values that we can make whatever interpretation that we would like to do, and that will be. So we also have to keep in mind that the curve fit should also be proper. Okay, so this one is not quite where it is, but this is more or less okay. Okay. So in addition to that, there is, because I talked about the optimization experiments. So obviously that is something that you have to it can be done manually, but also this program offers a automated way or a tool that can be optimized to use this. So this is called ITC experiment design. You can just call it from the previous menu, and then here whatever that first run that you've done, use the data from here, and then I will give the all the parameters, and then I will just simply hit optimize. Then it will look for the obvious concentrations that it will give, and then throw out the desired concentrations here. So what is the concentration of the inhibitor should be, and what is the concentration of the enzyme should be. So usually what the rule of thumb is that whatever the enzyme concentration that we maintain at the cell, the inhibitor should be at least tenfold higher than that. Okay, so that is what we use like that. So with this tool, help of this tool, you will be able to, the optimization would be done seamlessly with the one run of it, because otherwise we might have to, without this tool, then we have to do a few runs to ensure that we have proper concentrations. Okay, now this is also helps. So this is pretty much what how we can run the experiment and analyze the data, and we'll get the exact parameters. So obviously this one does not list the delta G values, but delta G values, obviously, as I said earlier, it can be calculated, and that is not something that we usually report. Okay, the only difference that we require is the relative contributions of delta H and delta S that is used for the this thing, and then obviously KD that has affinity, and then N is the stoichiometry. Okay, so that is what it is. So for a case study, I'm going to look at one of my publications that is about 5 or 6 years ago, whereby I try to characterize the binding mode of one of the enzymes. Okay, so this work is published in JACS in 2012, I think. Okay, so the protein of interest is the mitotic kinase in EG5 that has is a validated cancer drug target, and with the many of its compounds in various phases of clinical trials. And one of the inhibitors that we try to design that is called the BI8 is known to bind in two binding sites. Okay, so we already know there are several inhibitors that has been structurally characterized, functionally characterized, that are monostrol, ispinazib, STLC, and so on. So I'm going to use some of these inhibitors here. So the monostrol, ispinazib, both of them bind to this site 1, that is this point here, that is where site 1 I refer to as site 1, it binds here, and the BI8 is proven to bind at these two sites. So the BI8 is inhibitor that were reported in this article, and it is weak binding obviously, and these two binds to two sites. Okay, and the monastrol is also weak binding, while ispanazib is a strong binding inhibitor and is one of the clinical drug candidate. Okay, that is being done, and it is proven that monastrol and ispanazib are known to bind binds to site 1 in 1:1 ratio, and BI8 binds to both the sites. You would like to study that into further. Okay, so we carried out various ITC experiment using microcal ITC, and these are the results that we have carried out. So I'm just going to point out the stoichiometry to prove that the new inhibitor BI8 binds to the same binding site as the ispanazib and the existing monastrol. Okay, so as I already mentioned, monastrol is a weak binding inhibitor. Ispanazib is a strong binding inhibitor. So when I titrate ispanazib with EG5, I get a molar ratio of stoichiometry of 1:1. And when I add monastrol to this ispanazib EG5 complex, there is no binding because monastrol is a weak binding inhibitor and hence cannot replace ispanazib. When I reverse it, it actually does not happen that way. So when I reverse it, that is when I titrate monastrol first, obviously as it is being a weak inhibitor, still it has 1:1 binding stoichiometry. But when I add ispanazib, titrate ispanazib after to the EG5 monastrol complex, then ispanazib will replace monastrol because it being a strong binding inhibitor having higher affinity towards EG5, it replaces monastrol. So thus this is that proves ispanazib can replace a weak binding inhibitor that is bound to EG5 in site 1. Okay, so we carried out the ITC studies on BI8, that is the compound of our interest, and it is proven by the ITC. It shows a stoichiometry of 2, and it says it is a sequential binding of two sites. That means that it goes and saturates at sites 1 and then move on to the site 2. So that is a model that is well fit, and it gives you the data. Okay, so now what I did is we injected the ispanazib after this. Okay, so ispanazib being a stronger ligand as in the case of monastrol, it did the same thing. So it did replace only one of these sites. So we still have only because it reacted with 1:1 stoichiometry. That means that it removed only one of the BI8. The other one bound at the second site is still remains bound. Okay, so so that tells us that BI8 binds to the existing site 1 where ispanazib, monastrol all binds, but the binding is weak in such a way that BI8 is binding. BI8 is weaker. Okay, and if we reverse the experiment, that is you just the same as this one. So we saturate the EG5 with ispanazib, that is meaning that we block the site 1 so that we cannot access it. And then when you inject it with the BI8, now it comes up with the one site occupancy. So the stoichiometry is only 1. So that means it binds to the site 2 alone, because site 1 like monastrol, it cannot replace. Obviously, there is no binding. So site 1 is fully occupied or blocked by ispanazib, so it cannot bind, and the BI8 binds only to the second site, which ispanazib does not bind. Okay, from these experiments, I can say that BI8 binds to two binding sites at the stoichiometry of 1:2. So the one of the site is same as the existing site, that is ispanazib, monastrol, all of them binds to one site, and that is what it is done. Okay, the second site is different from this one, hence it is unique, and it is not where ispanazib and other inhibitors bind. So that means that we have identified a new binding site, and this particular observation is further confirmed by the structural studies. So in the absence of structure by this experiment, I could tell that one of the binding site is common or the existing binding site, and the second one is new site. So likewise, in the absence of structure, this would have been a conclusive proof for that hypothesis. So like this, there has various applications for ITC, and if you have any questions, please contact me for that, and I hope this video would be useful for you. Thank you for listening. Thank you for watching. Thank you. Good luck.